Hello Bandhugan, Namaskar, welcome back to my YouTube channel Sahil Educations. If you are studying biology and if you are really interested in the subject, then you will definitely encounter some questions upon the muscle physiology. That is, what is sarcomere and what is the structure of sarcomere. So let's start talking about the sarcomere today. If we start studying the sarcomere, then we'll have to start from the muscle fiber, which is the cell of our muscle. Definitely each and every organism is made up of cells that becomes tissues and then organs and then the organism. And in case of muscles also, you will see some cells. These cells are called muscle fibers. Okay, these muscle fibers do contain numerous muscle fibrils or myofibrils. It can be called myofibrils. Okay, just elongate one. This one is the myofibril. It is also called as filament of this muscle fiber, and this muscle fiber is then covered with a membrane which is sarcolemma, and then along with this sarcolemma, this muscle fiber is again covered with a connective tissue. That connective tissue is endomysium. Okay, when you see this muscle fibers and then these myofibrils, you will definitely see it like this. If you elongate this one, you will definitely see some dark lines, consecutive dark lines. Okay, and in between these consecutive dark lines, you'll see some bands, dark bands. So this is the dark band, then light band, dark band, then light band. This all makes up this muscle fibril, and this muscle fibril, a bundle of muscle fibril makes this muscle fiber. So you can see a muscle fiber like this. Okay, so in case of sarcomere, this one is the sarcomere, which is a contraction unit or contractile unit. Unit of contraction of a muscle fiber is called sarcomere. Okay, so then what is present in this sarcomere? Let's discuss about this sarcomere in details. If we draw a sarcomere, then there must be these two dark lines or dense lines. These lines are Z lines. These lines are Z lines. Okay. In between these two Z line, there is another light line, very thin and light line. This line is the M line. As because this is present in the middle of these two Z lines, so this is M, middle. Okay. Then you will see some thick filaments, microfilaments actually. So some thick filaments, microfilaments. These thick microfilaments are myosins. What are these? These are myosins. Okay, which runs in the both side of this M line, the central M line. Let's talk about the actin microfilaments now. These actin microfilaments look like very light and thinner microfilaments, and these thinner microfilament will be present in the both side of this Z line. In the both side of this Z line. These actin filaments will definitely incorporate in between these myosin microfilaments. This actin runs deep inside to the myosin microfilaments. Okay, now talk about some bands present here. Some kind of band you can see in this the structure of sarcomere. this is the Z line, and beside these Z line there are two bands. This one is the band and this one is a band. These bands are I bands. These bands are I band. Okay. After this I band, you can see alternatively the myosin, actin, myosin, actin, myosin, actin, myosin proteins or microfilaments are present. This band is the A band. Just the alternative actin and myosins are present in this band. In case of I band, only actin is present in case of I band. Okay, this is the basic difference between I band and A band. What about this next band? This is called Z band, H band. Sorry, this is called H band. This H band only contains the myosin microfilaments in this sarcomere. So let's see the bands in this side also. This one is the I band, which only contains the actin microfilaments. In case of A band, which contains the myosin and actin alternatively, and in case of H band, this only contains the myosin microfilaments. So this is the basic structure of a sarcomere, which is a unit of contraction in case of our muscle fibers. So when muscle contracts, these two Z lines comes closer. When these two Z lines comes together or comes closer, then the muscle has contracted. And if these Z lines goes apart from each other, then it will be called as relaxation of the muscle. So apart from these, the I band, H band, A band, M line, Z lines, there are certain proteins which actually defines the length of this actin and myosin microfilaments. So let's talk about these proteins also. In case of actin microfilament, these are the proteins, and some proteins are also present here to define the length, to accurate the length of this actin microfilaments. So in case of actin, there are alpha actinin, tropomodulin, and cap Z. These three proteins are actually present to define the length of this actin microfilament in the both side of this Z line. It has been said, or it can be said, that this alpha actinin is basically present in the both end of this actin microfilament. Okay, so it can control or it can regulate the length of this actin microfilament. Okay, this alpha actinin is bind with this M line. This alpha actinin is bind with M line, so that the actin microfilament do not come excessively inside this myosin microfilament, or it cannot touch the M line. Okay, so this is alpha actinin for you. And in case of tropomodulin and cap Z, these are present here in the Z line. In this Z line, they are just hanging. In case of tropomodulin and cap Z, just these proteins are present in the mid of the Z line, and they are just hanging or holding the actin microfilament in the both sides, so that they do not fall down. Okay, so this is the basic concept behind these proteins presence and the actin microfilaments. What about this myosin? These are the heavy proteins, the thicker proteins. Okay, so there are some protein like titin, myomycin. These proteins are present to control the length of this myosin microfilament and to hold this myosin microfilament in the two sides of this M line. So titin is present like this. Titin is actually defining the length of this myosin microfilament. It actually binds the Z line with this myosin microfilament, so that myosin do not enter excessively into these I band. Okay, so this is the function of titin. And in case of myomycin, this myomycin is present here so that it can hold this myosin microfilament in the both sides of this M line. So this is all about the structure of sarcomere. You can have the question about the sarcomere in different forms, in different languages, but the answer will be same. If you are going to be asked as elaborate the structure or define the structure or write a short note upon sarcomere, then these things should be written in your exam copy. So all the best, and if you like the video, please do comment, and if you have any doubt upon this structure of sarcomere, do let me know in the comment section. And thank you, all the best. See you in the next video. If you find any problem in memorizing these bands and lines and its names, then you will apply a trick. The trick is like 0 is actually home made. Here home means India. Definitely I'm Indian, so this is home, and 0 has been invented in India. So this Z stands for the Z line, I stands for this I band, A stands for this A band, H stands for this H band, and M stands for this M line. So this is the sequence, and you will remember it or you will memorize it in a way like 0 is actually home made.